leveling up with that gram-negative bacteria. With that PPT, I have sprinkled with some more 2025 updates from that World Health Organization and that CDC to keep that real fresh. In that nursing squad to dog that superbug sketches. If it is a battle to resist the trench and slide by slide to zip in that casual coverage of the jam, it is a 5-player focus in that casual vibes. With that, I am Sornambiga, SNS College of Nursing. Today I am going to talk about the topic about gram-negative bacteria from microbiology. In the gram bacteria, negative bacteria, it is a title, it is a subtitle points is also we have to spotting out with its own and throwbacks at still top and villains. We have to new amounts also. It's this kind of pumps you have to level up and go. What is that gram negative bacteria? It is a healthcare is most processing and evidence based institutional strategies of this. That understanding the gram positive or gram negative bacteria is very tough. In the gram negative is a structural characteristics should say it is a skimming and peptidoglycan layer. It is a so thin peptidoglycan layer present in that. That outer member layer is jumped with that LPS. In that LPS is nothing but lipopolysaccharides. It is a dish, it's a purple pink gram taste. Total that it is a shield protection of that. Make a hot kill to know on that. In the 2025 scope, it is wall to got 2 to 3 peptidoglycan layers or there present in the gram negative bacteria. In that antibiotic resistance to global headaches, which is in that critical pathogens to prove, like E. coli causing UTI to septic escalators. In that Klebsiella causing respiratory and bloodstream infections. In that Pseudomonas aeruginosa, so it's a opportunistic multi-organ infections also. In that Acinetobacter causes the hospital acquired infection. So always that gram negative bacteria causing the headache to the hospital environment to always causing the disease also by the way of lipopolysaccharides. Moving on to that identification. How we are identify the bacteria by the way of gram staining method. It is one of the basic and easiest method to stain the bacteria by the way of culture test to then McConkey growth medium. It is a pink light gram negative first IDs with the bug of clubs. In the times, we have to learn about the bacterial cell wall structure. It is the empirical antibiotic therapy to ensure the proper treatment. And we have to select the specific media to grow the gram negative bacteria. In that molecular methods like a PCR and a sequencing, also we can able to by the way of method we can identify in that bread culture to super speedy recover to the lab goals. Moving on to the virulence and resistance mechanisms. In that virulence and what is virulence and what is resistance? Virulence is that nothing but it is a disease causing capability of the bacteria. It is outer membrane contain lipopolysaccharide. Already we saw this. In that potence, that exotoxins and that endotoxins to trigger the severe immune response to that leading septic shock to the multi-organ failures in the vulnerable patients. And that enzyme production is the beta lactomases is the specific condition to produce the virulence to the bacteria. It is overall against to the antibiotics. And then efflux system, in that efflux system is nothing but the bacterial cells most probably reduce the drug's permeability and then it is caused a treatment to the success for the patients. Moving on to the clinical impact. What is clinical impact and infection control? It is the bacteria, it is divided into fraction of second in the thousands. So we have to prevent as a fast manner also. Clinical impact is controlled to that syndromes glare. It is the ICU, catheters, UTI sepsis and the blood roggers and wound fist, meningitis and immunoweakened patients also. They can also suffer from these diseases easily. And then infection control, we can able to control the infection by the way of sanitizing and that sterilizing procedures also. Next, moving on to the novel treatment. What is novel treatment? In these cases, we can able to treat the bacteria by the way of therapy method, novel combinations, stewardship protocols, and prevention strategies. In this method, we can able to prevent the patient by the way of treatment or as a sterilizing procedure or as a sanitizing procedures also. And to that integration strategies with them to educate and training the patient. In these cases, the continuous professional development also and that staffs also can prevent the gram-negative bacteria in the biological method, and diagnosis capability is then important to implement the rapid gram staining method. Nowadays, we can able to by the way of gram staining method to the patient in that fraction of second by the way of AI tools also. Then infection control is a protocol, it's an isolation procedure, environmental surveillance, and outbreak response to the mechanism, and antibiotic stewardship. That antibiotic stewardship is the therapeutic drug to give the patient to recover the medicational purposes. And research and innovations to activate the participant in the clinical trials, surveillance network, and development of the novel antimicrobial and diagnosis tool to the patients. Case study here I enclose one small case study for your reference purposes. In 2024 in ICU experiments to significant outbreak of the multi-drug resistance like a Klebsiella pneumoniae. It is nothing but pneumonia, fever or else pneumonial bacterial infection affecting the 30 patients at the stage with the consenting mortality rate of the 25%. What they are identified from that cases, it is a to prevent, educate the staffs and educate the patient to sanitize thoroughly, and after the 6 months it is comprehensive invention to 60% of cases reduced from these challenges. To that visual summary of the gram-negative bacteria is the gallons of unique structure. It is comparatively acid-fast bacteria and gram-positive bacteria. It is a unique structure and it is a one type of clinical pathogen. It is a critical pathogens like a Escherichia coli, Klebsiella, Pseudomonas, Acinetobacter is a severe life-threatening infections. After that, it is a response to that education to the patient and diagnosis, infection control, stewardships, and that research initiative we have to follow in that methodology. Overall, what we are taken from home. In this understanding to apply the comprehensive five-pillar concept to absolute essential and compact growing through the bacterial world. It is continuous education to the patient and to the healthcare workers. Advanced diagnosis method. In this advanced diagnosis method is one of the most popular method nowadays. We are following in that categories of that. And prudent stewardships. In the prudent stewardships, we have to give the medication to the patient and give the awareness to the patients also, and innovation research. We have to give the innovation research. Thanks for watching. If you found this session useful, do like, share and subscribe for that more microbiology insight.